lapel.
Welcome to the deep dive. Okay, so if you're deep in preparation for that PhD qualifying exam in pharmaceutical sciences, this one's really tailored for you. We're focusing today on human drug metabolism.
That's right. Specifically, we're going to unpack a really critical piece, the cytochrome P450 enzymes. You know, the CYPs.
Exactly. And our mission today isn't just about listing facts. We want to help you really get these major concepts, break them down so they're easy to remember, easy to explain.
Yeah, the goal is for you to feel confident, like you could stand up and lecture on this yourself. Understand the core pathways, the key players.
So you can articulate the implications, the clinical relevance with real clarity.
And honestly, getting a handle on these enzymes, their substrates, what blocks them, what ramps them up, it's just fundamental beyond any exam.
Right.
It's how you start to really predict how drugs will behave, how they'll interact.
Yeah.
It's about safety, efficacy, the whole picture. This dive should give you some solid frameworks.
Crucial stuff. Okay, so let's get into these individual CYP isoforms then, these enzymes that are, constantly working away in the body, breaking down drugs, other compounds.
And CYPs, remember, are this huge superfamily, absolutely key for biotransformation. Often, their main job is making things more water-soluble.
Easier to get rid of.
Exactly.
Yeah.
So your body can excrete them. We're going to focus on the heavy hitters, the most common, most clinically relevant ones you absolutely need to know.
Right. Let's start with CYP1A1 and CYP1A2. I gather these are pretty involved with environmental stuff.
That's a good way to put it. CYP1A1, it mainly goes after polycyclic aromatic hydrocarbons, PAHs, and some organochlorine insecticides.
Okay.
Inhibited by something called naphylflavone. And it gets induced, interestingly, by the PAH themselves, also smoking, omeprazole, and those organochlorines.
And 1A2.
1A2 has a broader palette, you could say. It handles drugs like amitriptyline, imipramine, tizanidine, caffeine, clozapine, haloperidol, mexaltine, ondansetron, propranolol, terracrine, theophylline, verapamil, arwarfarin. More PAHs too. It's busy.
Wow. Okay, what about things that slow it down or speed it up? Inhibitors, inducers.
Sure. For inhibitors, think amiodarone, cimetidine, furifilene, fluvoxamine, diclopidine, ciprofloxacin.
Got it.
And the inducers are really interesting because they tie into everyday life. You've got PAH amines, like from barbecued or flame-broiled meat.
Seriously, barbecue.
Yeah, also Brussels sprouts, broccoli, those cruciferous veggies. Insulin, tobacco smoke, omeprazole again, phenytoin, modafinil. Smoking's a big one.
That's fascinating. It really connects diet, barbecued meat, broccoli, and lifestyle like smoking directly to how you metabolize certain drugs via CYP1A2. Huge real-world relevance there.
Absolutely. Before you even get to the genetics, the environment plays a huge role.
Okay, so from those environmental sensors, let's shift to CYP1B1. What's its story?
CYP1B1 is interesting. It handles a mix, things like tamoxifen, more PAHs, aromatic amines, aflatoxins, which are nasty toxins.
Right.
Docetaxel, an anti-cancer drug, and even estrogens.
So endogenous and exogenous compounds. What inhibits or induces it?
Inhibitors include some PAHs like pyrene, stilbene, certain flavonoids and coumarins, resveratrol, the stuff in red wine, mitoxanone, flutamide, taxols.
And induction.
Induction comes from things like PCBs, Aerochlor 1254, it's a classic example, naphthoflavone, and again, PAHs from smoking.
So when you're thinking about this for your exam or just understanding its impact, knowing 1B1 metabolizes tamoxifen, crucial for breast cancer, and also those potent aflatoxins really highlights its importance in both therapy and toxicology. It's a key player.
Definitely.
All right, let's group the next few. CYP2A6, 2B6, and 2C8. Distinct roles, but all important. Walk us through them.
Okay, so CYP2A6, substrates include coumarins, aflatoxins again, nicotine.
Nicotine. Big one.
Huge one. Also tigafer, efavirenz, valproate, halothane, 1-ron-3-buteti, and letrozole. It's inhibited by phenylcypramine, methoxyline, and surprisingly, grapefruit juice.
Grapefruit juice again. Okay, inducers.
Phenobarbital and rifampicin. Pretty standard inducers.
Got it. What about 2B6?
CYP2B6 handles things like bupropion, the antidepressant, and smoking cessation aid coumarins, cyclophosphamide, mephenetoin, methadose. Yeah, methoxychlor, which is a pesticide. PCP, ketamine, and efavirenz again.
And it's inhibitors and inducers?
Inhibitors. Tranylcypramine, thiatepa, diclopidine. Inducers. Phenobarbital, rifampicin. Efavirenz itself actually induces it. Modafinil and methoxychlor.
Self-induction with efavirenz, interesting. And 2C8.
CYP2C8. Substrates include imodiaquine, an anti-malarial cervastatin, which was a statin pulled from the market.
Right.
Paclitaxel, tolbutamide, montelukast, enzalutamide, repaglinide. Inhibition comes from quercetin, glozone drugs, gemfibrozil, especially its glucuronide metabolite.
Okay.
Sequenavir. Induction again. Phenobarbital and rifampicin.
So looking at these three, you really see specific clinical impacts, like 2A6 and nicotine metabolism key for understanding smoking dependence and cessation.
Exactly.
And 2B6 with methadone and bupropion directly relevant to addiction treatment and psychiatry. These aren't just abstract pathways.
Not at all. They have direct patient consequences.
Okay, moving to another crucial pair, CYP2C9 and CYP2C19. These seem to pop up everywhere with common drugs. Why are they so central?
Well, they metabolize a huge number of widely prescribed drugs. Take CYP2C9. It handles amitriptyline, dapsone, fluoxetine, fluvastatin, loads of NSAIDs.
Like ibuprofen, naproxen.
Yep, phenytoin, sulfonylureas for diabetes, tamoxifen again, and very importantly, S-warfarin.
S-warfarin. The more active form, right?
Critical.
Absolutely critical given its narrow therapeutic window. Inhibitors include isoniazid, fluvastatin, fluvoxamine, lovastatin, sulfenazole, diclofenac. Inducers are secobarbital, rifampicin, enzalutamide, elvitegravir.
And 2C19.
CYP2C19 is also vital. Think barbiturates, citalopram, mefenitoin, proton pump inhibitors, PPIs like omeprazole, lansoprazole.
Very common.
Extremely common. Also, phenytoin, proguanil, R-warfarin, clopidogrel, the antiplatelet drug, and voriconazole, an antifungal.
Clopidogrel activation, right. That's a big one. Huge. It's a prodrug that needs 2C19 to work. Inhibitors include tranylcypromine, cimetidine, fluoxetine, ketoconazole, diclopidine, fluvoxamine. Inducers are carbamazepine, norethindrone, prednisone, rifampicin, enzalutamide.
The clinical significance here is just massive. 2C9 and warfarin, tiny changes in metabolism can cause major bleeding or clotting risks.
Precisely.
And 2C19 with PPIs and clopidogrel. Whether someone gets heartburn relief or is protected from a heart attack can depend heavily on this enzyme's function and potential interactions.
It really drives home the need for careful medication management and sometimes genetic testing.
Absolutely. Now, let's talk about CYP2D6. This one has a reputation for being highly polymorphic. What's that mean, and why is it so important in pharmacogenetics?
Yeah, polymorphic just means there's a lot of variation in the gene between individuals in the population, so people naturally have very different levels of CYP2D6 activity. Some metabolize drugs super fast, others very slowly, and most are somewhere in between.
And it handles a lot of important drugs.
A ton.
Yeah.
Especially in psychiatry and cardiology. Think tricyclic antidepressants, TCAs, many antipsychotics like haloperidol, antiarrhythmics like flecainide and mexiletine, beta blockers, timolol, S-metoprolol, buforolol, MDMA or ecstasy, SSRIs, opiates like tramadol and codeine, converting them to their active forms, and venlafaxine.
Codeine activation, that's a classic 2D6 example.
Classic. If you're a poor metabolizer, codeine basically doesn't work for you because you can't convert it to morphine efficiently. If you're an ultra-rapid metabolizer, you can get toxic effects from a standard dose.
Scary. What inhibits or induces it?
Inhibitors are common. Amiodarone, cimetidine, ranitidine, older antihistamines like chlorpheniramine, quinidine, and importantly, many SSRIs like fluoxetine and paroxetine can inhibit it. St. John's wort too.
So an SSRI could block the metabolism of another drug handled by 2D6.
Exactly. A potential drug interaction right there. Induction is less common, but pregnancy can induce it, as can rifamycins and corticosteroids.
The variability in 2D6 really underscores the need for personalized medicine, especially with antidepressants and opioids. It's central to understanding why people respond so differently.
Absolutely fundamental concept for your exams and beyond.
Okay, what about CYP2E1, often linked to lifestyle, right?
That's right. CYP2E1 substrates include things like benzene, the muscle relaxant chlorzoxazone, ethanol, alcohol.
Ah, alcohol.
Yep. Flaring anesthetics like halothane, and crucially, paracetamol, or acetaminophen.
Paracetamol, okay.
It's inhibited by sulfides, like disulfiram, the drug used to treat alcoholism, diallyl sulfide from garlic, for instance.
Yeah.
And induction is key here. Induced by ethanol itself, acetone, the TB drug isoniazid, starvation, and diabetes.
So the ethanol induction, what's the clinical takeaway there, especially with paracetamol?
It's a major issue in toxicology.
Oh.
Chronic alcohol consumption induces CYP2E1. Paracetamol is partly metabolized by 2E1 to a toxic intermediate. So if you have induced 2E1 from drinking, taking even normal doses of paracetamol can potentially lead to more toxic metabolite formation and increased risk of liver damage.
Right. A really important interaction to counsel patients about.
Definitely.
Okay, let's wrap up the enzyme tour with the big one, or rather the big pair. CYP3A4 and CYP3A5, often grouped together. Most abundant, most versatile.
Absolutely the workhorse. CYP3A45 metabolize a staggering percentage of drugs on the market. It's estimated maybe 50% or more of clinically used drugs are substrates.
Wow. Like what?
Oh, the list is huge. Aflatoxin B1, again, older antihistamines like terfenadine and estomazole, calcium channel blockers like philodipine, cannabinoids, cyclosporine, macrolide antibiotics like erythromycin,
immunosuppressants, antibiotics,
opiates like buprenorphine, fentanyl, sufentanil, protease inhibitors for HIV like ritonavir, most statins except pravastatin, tacrolimus, paclitaxel, midazolam.
Midazolam, the classic probe drug.
Exactly. The Sazodone, dasatinib. It just goes on and on.
Incredible scope. So inhibitors and inducers must be really important too.
Hugely important. Inhibitors include amiodarone, azole antifungals, ketoconazole, fluconazole, voriconazole are potent inhibitors. Cimetidine, grapefruit juice.
The famous grapefruit juice interaction.
That's the one. Primarily a 3A4 inhibition effect in the gut wall. Also macrolides, endogenous steroids, protease inhibitors.
And inducers.
Also a long list. Barbiturates, carbamazepine, glucocorticoids, glitazones, nevirapine, phenytoin, rifampicin, the classic broad inducer St. John's wort.
St. John's wort again.
Yep. Modafinil, mitotane, efavirenz, clotrimazole, enzalutamide, dicloxacillin, aprepitant, many potential interactions.
It really drives home how vital understanding 3A45 is. Because it handles so many drugs, interactions are incredibly common and can seriously affect outcomes, either making drugs ineffective or toxic. That grapefruit example is perfect because it's so common.
It's a textbook example for a reason. Simple dietary choice, major metabolic impact.
Okay, so that's a whirlwind tour of the major CYPs.
Yeah, and the key takeaway, especially for your exam prep, is really getting a grip on the main substrates, the key inhibitors, and the most common inducers for each of these major isoforms.
It's not just memorizing lists though, right?
Not at all. It's about building that mental framework. If you know drug X is metabolized by 3A4, the patient starts drug Y, which inhibits 3A4.
Yeah.
Ding, ding, ding. You can predict a potential interaction. You can start to explain why responses vary, why toxicity might occur.
Right. It's about connecting the dots.
Yeah.
Knowing these specific examples we discussed, understanding the implications, that shows a deeper grasp than just rote learning. You see how these pathways interconnect and impact real patients.
Exactly. It's foundational knowledge.
So to help you lock this in, maybe we can run through a few quick scenarios, kind of like self-test questions you can use.
Good idea. Okay, how about this? 1. Think about a patient on clopidogrel. Which main CYP is needed to activate it? And can you name maybe 2 common things, drugs or substances that could inhibit that enzyme and potentially make the clopidogrel less effective?
Okay, good one. How about this?
2. Picture someone who drinks grapefruit juice every day. Which major CYP gets inhibited most significantly? And what are, say, 3 classes of drugs whose levels might dangerously increase because of that inhibition?
Number 3.
3. Okay, codeine for pain. Which CYP converts it to its active form? And what happens clinically if your patient is a poor metabolizer of that specific enzyme? What would you expect?
Right, thinking about the consequences. What else?
4. Let's say you have a patient who drinks a lot of alcohol chronically, and they overdose on paracetamol. Which CYP isoform is likely induced by the alcohol, and how does that induction make the paracetamol overdose potentially worse?
Nice link between lifestyle and toxicology. One more.
Okay, last one. Can you list maybe 3 broad categories of drugs, think CNS or cardiovascular, that are often substrates for CYP2D6? And name 2 common inhibitors, maybe even over-the-counter things, that could cause levels of those 2D6 substrates to build up.
Excellent. Those should really get you thinking and applying the knowledge. And that brings us towards the end of this deep dive. Hopefully going through the cytochrome P450s like this has been genuinely helpful for your qualifying exam prep. Mastering these details really is a cornerstone of pharmaceutical sciences.
Absolutely. And maybe one final thought to leave you with. This whole area, the interplay between drugs, diet, genetics, it's incredibly dynamic. What we know about drug metabolism is constantly evolving.
That's true.
So as you prepare and as you move forward in your career, keep that critical mindset, keep questioning, keep integrating new findings. That's what transforms you from just a student into someone who really contributes to the field. Always more to learn.
A really powerful reminder. Keep questioning, keep learning.
PharmaPal.